Cool. So I'll move on to part 3. So this is assembling your sequencing data using command line interfaces. So you will have within each of your sort of barcode folders, or you can rename them as, you know, whatever TB1 or whichever isolate it is, if you know what it is. You will have so many FASTQ files, and now you need to sort of stick them all together and create this big jigsaw puzzle of your assembled genome. So there are different ways of doing this. As I mentioned, Minimap 2 is a really useful tool to do this. So it aligns DNA to a large reference database. So you can download a reference genome from GeneBank or wherever. The only problem really is that the requirements needed, you need at least 12 gigabytes of RAM to run it. So if you think about how it works, it's basically doing hundreds of different things all at once. It's got a reference genome, and it's taking all of your FASTQ files and trying to place them all, put them into the right place within the sort of jigsaw that is that genome. So it's, I think most standard laptops are about 8, so you might need to invest in a more high-powered computer, or there are cloud-based options as well. And you can use Linux or Mac to do align with Minimap 2. I won't go through actually running the code because, as I say, you will have hundreds of FASTQ files, and that would take me ages. So nobody wants to sit here and watch me wait for that. But it's the same principle in that you have the address to wherever Minimap 2 has been stored on your computer. You have the address to the reference genome file that you want to align it to. You have the folder where your FASTQ files are, and then you want to tell it where to align and what to call the output file extension. These here, so A just tells the program that you want to generate your alignment into a SAM format, and then X is an option to explain further options, which is this map-ONT, and this just basically means you're telling Minimap that you are using Oxford Nanopore data, and that there's potentially going to be noisy long reads, which means a higher error rate. So it's just basically explaining to it, just be careful, because what you're seeing might not necessarily be exactly what you think, because nanopore data is not necessarily as accurate as Illumina. We then have Flye. So if, as I said, you don't know what your organism is, or it's a very new thing that doesn't have reference genomes available, you can use Flye to piece it together. So if Minimap 2 is like having the picture of the puzzle on the top of the box, Flye is like piecing together the puzzle when it's upside down and you can't even see what the picture is supposed to be. So Flye, it's more difficult to do it this way, but sometimes you don't have the option, or sometimes there are advantages to doing it this way. So it's useful for long read, such as PacBio and Oxford Nanopore, and it's designed to be used for loads of different things. So from small bacteria, genomes, viruses, and also all the way up to large mammalian sort of size assemblies, which are going to be much, much bigger. And it takes all of your data, so it takes your reads and outputs them into contigs. So as I say, often if you bases in your FASTQ files to cover the entire genome, and so what it does is it puts together these sort of islands called contigs, and it sort of says, right, well, I think this sits here, but then I've got a gap. And so within your file, you will have these different contigs. Obviously, the more sequencing you do, the less or the more coverage of the whole genome you get, and the more or the less but larger contigs you should have. And it also has a mode for metagenome assembly as well. Again, you need 12 gigs of RAM. You can use Linux or Mac to run Flye. So here is the basic code again to use Flye. So Flye is the address where Flye is stored, or as I say, you can link to it. You've got your number of threads again. So nano-raw, this is specifying to Flye that your data is Oxford Nanopore and you processed it using Guppy. And again, if you look on the Flye website, it will explain all of the different commands and it will explain which ones you need to use depending on what data you've got. This is where your FASTQ files are. So this is the ones you want it to sort of combine together. And this is the estimated size of your genome. I don't think you have to put it, but it's if you have a vague idea of how big you think your genome is likely to be. So for instance, if it's a bacteria, you can give it kind of, it's just a guideline to help it try and work out the best. Because if it's assembling something that it thinks is mammalian, but assemble it in a different way. And then this here is where you want to output your data to. Great. So then, as I mentioned, Oxford Nanopore data, because it's not necessarily as accurate, you might need to polish your assembly or your contigs. And Madaka is a really useful tool. It's useful in, it has lots of different functions, but it's helpful to polish your assembly to try and make sure that it's as accurate as possible. So the two uses I have for it is polishing, and you can also use it for variant calling. And again, you need at least 12 gigs of RAM, and you can use Linux or Mac to run it. So Madaka, you have to activate the Madaka environment to run it. So if I just show you, I don't want to, okay, that's fine. So you can see here that base, so that's, I am within sort of the base environment. But if I changed it to hopefully conda activate Madaka, oh, that's not what I asked. Except I think mine is 2. Yes. It will then change it to the environment for Madaka, and that just basically means that that environment has all of the right sort of programs and files and things and setup that Madaka needs to run. Mine is 2 because I accidentally made 1 and it wasn't the right one, but you shouldn't have the same problem as me because I was messing around. That's how you activate Madaka. And then this is the code for polishing it. So you've got Madaka consensus. So you can also do Madaka variant calling, but consensus is the polishing tool within Madaka. So directory is the folder or directory where Flye or Minimap 2 has outputted your assembly file to. So that's just the input address, as it were. And this is the specific file that you need or that you're wanting to polish. This is where you want to put it. So it usually creates a folder called Madaka within whichever folder you've put it, you've told it to put it in. Back to threads, so you can change that. And then this is the model. So again, you can see here 9.4.1, that's the flow cell. But again, you need to check on the Madaka website to make sure that you're using the correct model for the type of hit and whatever that you're using. So I think it still works, but you'll probably get, your data might not be quite as accurate. Cool. Does anybody have any questions about that? It's a bit of a whirlwind tour. Is anyone still with me?
Yes, we're still with you.
Yeah, it's here. It's good.
I will probably have more questions for you during the week when trying this out.
Sure.
So
that's fine, as long as I haven't sent you all to sleep.
You're making it look so easy.
This I spent an entire week. I didn't come to work. I spent an entire week just sitting in a corner trying to code, and it was intense and hardcore. But do you know what? Once you've got over the first little tiny peak of not having a clue what you're doing, it's not so bad. So if you are at that stage, do not fear. I might be making it look easy, but I do not think of myself as an expert. As Sylvia knows, she is far better at this than I am. So anyway, if nobody has any questions about that part,
Lindsay,
that bit, once you've done that, you've got your lovely, oh, sorry.
There is a question in the discussion. So Jean,
oh, yes.
Flye is not making any assumptions of the data since it is de novo sequencing. Is that right?
So yeah, Flye, apart from what you tell it to make, like the size, it's not, yeah, it's basically just going along and fitting things together. So if you have a decent amount of depth, and in the PDF, I've explained the difference between coverage and depth. So coverage is the amount of your genome that you actually have data for. Depth is the amount of times that each base is has been sequenced. So depth is really important for this, because depth, a good depth means that if it actually is an A in your sequence and you've only got 4 versions of that, and it's an A, a T, an A, and a G, then you're not going to be very sure that that is actually an A. But if you have a coverage, a depth of say 30, and it's A, T, G, then you're going to be more sure, or the computer is going to be more sure that it's definitely an A. And so depth is also really important when it comes to fitting things together in a de novo way, because if you've got lots of versions of each section of DNA, then it's easier for it to piece it together. Because if you just have one and it's a small section, and the computer has to work harder, I guess, to try and identify where that fits in. But if you have multiple versions of sections of DNA, then it's easier for it to piece it together, I guess. So depth is really important, and that's why trying to get as many reads as possible for each of your sequences is really important. And I think it depends what you're doing, but having a depth really of at least 30 or 40 is probably the minimum that you should really be looking for if you're trying to, as I said, it depends what you're doing. If you're looking for SNPs, individual bases, then you're going to want to identify what species it is. Then you obviously don't need as much, because it will be easier to identify species. And so, yeah, Flye makes the assumptions that you tell it to make, basically. And so things like the genome length is just trying to help it, I guess, to identify. As I say, I don't think you have to put it in. If you have absolutely no idea, I guess if maybe you were doing, say, a sputum sample and you had no idea what you were looking for. Does that kind of answer the question?
Yes, thanks, Lindsay. It does. Thank you.
Excellent. Thank you, John. Does anybody have any other questions about that, or we can move on to downstream? No,